The session is about novel approaches for lymphocyte engineering, and I have to admit all topics have an interesting twist towards clinical application. And we will start with Dr. Michael Taxelmeier from the University of Vienna, who will talk about how to control the activity of CAR T-cells once they are infused back into the patient. Indeed, CAR T-cells are very potent therapeutic drugs, and sometimes they are even too activated in the patient's body. So what he proposes is a novel design of a CAR construct that is composed of a lower affinity binding domain that in its monomeric form is not capable of properly stimulating the engineered T-cells. However, upon the administration of a small molecule, the CAR can dimerizes, and in this state they have enough avidity to proper T-cell activation. So then we will move to the topic of T-cell product manufacturing, which is still challenging in terms of toxicity for the T-cells and safety and also cost. And in these regards, Dominika Bardeka from the University of Ghent will show with us the recent finding about a novel technologies that allows cellular transfection. And this technology is named laser-based photothermal, and she could demonstrate that by this technology she can achieve very high transfection efficiency with the minimal expenses of cell viability, which is very important. The third e-poster will tackle actually the problem of the limitation that we have in terms of preclinical models useful for studying the interaction between human tumors, human immune cells, and eventually also adoptively transfer human T-cell products. And here Juliane Mais from the University of Zurich will show her new model, where essentially she is using humanized mice that are already reconstituted with the human immune system, and these mice are further engrafted with autologous lymphoblastic cell lines. And very surprisingly, these tumors can be completely infiltrated by human immune system, and thereby we can study this interaction in a way that was not possible before. And then the section will be closed by Professor Katrin Schumann from the Technical University of Munich, and she will talk about regulatory T-cells, which is a subset of T-cells particularly interesting because of their unique role in controlling immune homeostasis. And I have to be honest, this cell population has been a bit neglected by adoptive T-cell therapy field in the last years, but holds a very huge potential in terms of therapeutics. And Katrin Schumann, by applying a CRISPR-Cas9 based screening, she discovered really important nodes of regulatory networks that govern Treg phenotype. And thereby she can provide really insights and important information to modulate basically Tregs according to a particular application.